Hello, this is Mr. Wartsky, and today we are going to start recombinant DNA technology. This is the first screencast. Now, if you have any questions, feel free to watch the video over again, refer to your textbook, or ask me in class. But let's get started. So we are going to be able to discuss how we can manipulate DNA, how we can do this, and why we do this. So recombinant DNA technology is broken down to the following topics: methods of obtaining DNA, tools and technology, production of protein or protein production, and uses. Today we hope to get through methods of obtaining DNA. So the first way that we can obtain DNA is called gDNA. G stands for genomic. So it's genomic DNA. And what we do is we cut the DNA from a cell's genome. In order to do that, we need chemical scissors to cut out the DNA. And those scissors are called restriction enzymes or restriction endonucleases. Those are synonymous. So what are these enzymes and where do they come from? Well, they come from bacteria. They are a way for the bacteria to defend against viral infections. What happens is the virus will inject the viral DNA into the cell, and these restriction enzymes will cut the viral DNA at specific locations. Those specific locations are called recognition sequences, and each restriction enzyme has its own recognition sequence. These enzymes were discovered back in the 1960s, and there are more than 150 different restriction enzymes. Now, they can cut in one of two ways. They can cut in something known as a 5' overhang, or sticky ends, or they can cut in a blunt end fashion. And let's discuss that in a second. So one restriction enzyme is called EcoR1. EcoR1 was isolated from E. coli, that's the ECO, and R stands for restriction enzyme, and this was the first one that was ever isolated, so it's 1. It's not EcoRe, it's EcoR1. EcoR1 cuts at a specific sequence of GAATTC. So every time there's a GAATTC, the restriction enzyme will cut after the G on both strands. Now, this is known as a 5' overhang because the 5' end here, see, has an overhang. It's also known as a sticky end because if you get two of these 5' overhangs together, they bind together very quickly. Here's an example of other 5' overhangs. We have HIN3 from H. influenzae, and it cuts at this sequence, AAGCTT, cuts after the A, and you'll notice there's a 5' overhang here. Or BAM3, which is B. amyloliquopheransins. Oh my gosh, that was easy for me to say. And it cuts at GGATCC, and it cuts after the G, and notice there's a 5' overhang there. So what does a blunt end cut look like? It cuts straight through. Like H. aegyptus cuts between the G's and the C's of GGCC, and EcoR5, which is from E. coli, cuts GATATC. It cuts after the first T. Right there. So these are blunt ends, and they don't join up, and it's hard for them to join up with other DNA sequences, not like the 5' overhang, which joins readily with other similar 5' overhangs. So now that you have cut out the DNA with these restriction enzymes, what do? Well, you put it into a probe because you want to produce the DNA through DNA synthesis, and you want to produce the protein through protein synthesis. The problem is, and does anyone see it? It begins with the letter I that prokaryotes do not have, but eukaryotes do have. Well, the I stands for introns. So you need to get rid of the introns if you're cutting the DNA out of a eukaryotic cell and putting it in a prokaryotic cell. And how do you do that? Well, that's the second way you can obtain DNA. It's called cDNA. C stands for complementary DNA, and basically it's the eukaryotic DNA without introns. Well, how do you produce this? Well, you need an enzyme that you might be familiar with called reverse transcriptase. And if you remember, reverse transcriptase takes mRNA and produces DNA. Now, the mRNA, as you know, has no introns and it has a poly A tail. So you would expose hnRNA to spliceosomes, and they sell kits to do this. You take the GNA, you expose it to RNA polymerase II, the nucleotides, and spliceosomes, and you will eventually produce mRNA. Now, how do you isolate the mRNA from everything else? Well, you need to think of the poly A tail. They have a, oh, there's what happens. You take DNA, sorry, and you produce mRNA, and then you're going to produce DNA again. So once you get the mRNA, you expose to reverse. How do you get it? Well, they have a poly A tail, and there's a film, plastic film with a lot of thymines on it. So the poly A tail will stick to the thymines when you wash it over the film. So then all you get is mRNA. Then you will expose the mRNA to reverse transcriptase and deoxynucleotides, and you will create a cDNA strand ready to be placed into a prokaryote. The third way that we can obtain DNA is by creating our own DNA sequences and chromosomes. We know about histone proteins, we know about telomeres, the centromere region. We can create small chromosomes with the DNA that we want and insert it into a cell. Usually it's a yeast cell, and we have done that, and they have acted normally. They are replicated, they divide, they behave just like any other chromosome would, except we created it. Another way to obtain DNA, and this is the fourth way, is through PCR. Now, this might be a little trick, but what we're doing is we're copying the DNA. We're amplifying it many times so that we can use it later. So we're not really obtaining the DNA, we're obtaining DNA copies. So PCR stands for polymerase chain reaction, and it copies the DNA. So we take the DNA that we want, we heat it up. The hydrogen bonds break. So the DNA strands separate. Now you have two single strands of DNA. If we cool the DNA and allow DNA polymerase to do its job with deoxynucleotides, we can synthesize the strand of DNA that we want. Basically, we synthesize primers, put them in with everything. The primer, remember, is the start sign, and DNA polymerase will start replicating from the primer on. We repeat this, and every time we do this, it takes 5 minutes. And we repeat this over and over again for 3.5 hours in a special machine known as a thermocycler. The problem is if you heat up DNA until the hydrogen bonds break in the DNA, when you do that, the hydrogen bonds in the DNA polymerase will also break and they will change shape. So that's not good. So what's the solution? The solution is TAC, and it is from the DNA polymerase from an archaebacteria called Thermus aquaticus. Thermus aquaticus is a thermoacidophile, and it's an archaebacterium, which means that it loves hot temperatures. So TAC will not denature at 100 degrees Celsius. We need primers, which we can synthesize in the lab, and we need a thermocycler that can heat up and cool down on a dime spontaneously. And we, of course, we need deoxynucleotides. And here's a diagram of how it works. We have the DNA sequence here. We heat it up. We cool it down to 72 degrees. The primers attached to the DNA. DNA polymerase synthesize DNA for a specific amount of time, and then it stops because we heat it up again. So we go from one strand to two strands to four strands to eight strands, and all the way over and over again. Every 5 minutes, another cycle runs, and we do that for 3.5 hours. We're going to start talking about tools and techniques today. So now we have the DNA, That's great, but now we need to separate the DNA so that we can use it. And we can separate the DNA by a process known as electrophoresis. Now, electrophoresis will separate the DNA by size and charge. You might be asking yourself, how? Well, we use a gel in gel electrophoresis, which is purified agar or agarose. Uh, we used to use acrylamide, but that's carcinogenic. So now we use agarose, which is from algae, purified from algae. The gel will separate the DNA by size. The small DNA can move through the gel a lot faster than large DNA, which moves slower. Now, electricity pulls the DNA through the gel because we know that DNA has a negative charge because of the phosphates. Therefore, DNA will be attracted to the positive pole of gel electrophoresis. How do we get the DNA into the gel? Well, we load it with something known as a digital micropipette, which we can use to load the DNA samples into the gel. And here's the setup. We have the power supply here, and we have an electrophoresis chamber here, negative, positive. Negative, positive. Yep. Now, we're going to include a buffer solution in the DNA surrounding it so that it will conduct electricity and it will help prevent the DNA from denaturing. When we're all said and done, we need to stain DNA because we can't see it with our eyes. We will end up with bands of DNA, and each band is a DNA fragment of the same size. We can cut the band out of the agar and use a solution called gel melt, which will melt the gel, and therefore we will have the DNA. And this is what a gel will look like after you stain it. You have a marker, which is a molecular ruler. These are of known size, DNA fragments of known sizes. And then we will have bands of varying sizes. Now, these are the wells where you will load your samples. This is the negative and positive end. These are the larger DNA fragments, and this is the smallest DNA fragment. We're going to stop there, and we're going to pick this up in the next screencast, which is screencast number 2. So again, if you have any questions, by all means, review the video or read your book or ask me questions tomorrow. Have a good night.